ربكما تكذبان فبأي آلاء ربكما تكذبان صدق الله العظيم. Ku berlindung pada Allah dari segala godaan setan yang terkutuk dengan menyebut asmamu ya Allah yang maha pengasih lagi maha penyayang. Wahai golongan jin dan manusia, jika kamu sanggup menembus melintasi penjuru langit dan bumi, maka tembuslah. Kamu tidak akan mampu menembusnya kecuali dengan kekuatan dari Allah. Maka nikmat Tuhanmu yang manakah yang kamu dustakan? Maha benar Allah dengan segala firman-Nya. Allahul muwaffiq ila aqwamit thoriq. Assalamualaikum warahmatullahi taala wabarakatuh.
Thank you, Mr. Muhammad Shafari.
Distinguished ladies and gentlemen, singing the national anthem of Indonesia, Indonesia Raya. Therefore, ladies and gentlemen, please rise for the national anthem. Bapak Ibu mungkin mohon untuk berdiri sejenak.
Ladies and gentlemen, moving on to the next agenda, that is report from the chairman of ICST 2023, Professor Insinyur Puan Anshari SD, MSc and PhD. So therefore, let us welcome him with a round of applause.
Bismillahirrahmanirrahim. Your Excellencies, Rector of Mataram University, Rector of Mula Warman University, Rector of Bina Nusantara University, uh, Dean of Faculty of Engineering, Director of Postgraduate Program, Head of LPPM, Head of LPMP 2, Head of LPPM Institute Technology Kalimantan, distinguished keynote speaker and invited speakers, distinguished participants, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Good morning. First of all, let's thanks to Allah Subhanahu wa Ta'ala and all praises to Him has given us mercies and blessing so that we can attend this meeting on the site the venue Raja Kuta Hotel Mandalika and join virtually from around the world and all part of Indonesia. Secondly, do not forget salawat and greeting always pour out for our Prophet Muhammad Sallallahu Alaihi Wasallam who taught their goodness. On behalf of organizing committee, it gives me a great pleasure and honor to welcome you all to the 8th International Conference on Science and Technology in 2023 in Hotel Rajakuta Mandalika, organized by Institute for Research and Community Service University of Mataram, University of Mula Warman, and Bina Nusantara University as co-host. It is a great privilege to address today as well reflect our journey leading up to this important event. Our theme for this year's conference is Sustainability in Research on Science and Technology for a better future. And I'm delighted to provide this report on conference preparation as chairman of this conference. For your information, in the 8th HST 2023, there are 6 keynote speakers, 8 invited speakers, 133 presenters. It consists of 32 participants come on site to present paper and 102 participants would present papers virtually. It's mean increase significantly in terms of the number of participants compared with the previous ICST last year, 2022. Ladies and gentlemen, I would like to express my deep gratitude to all keynote speakers, invited speakers, and presenters who submitted the paper in this conference. The presenters who joined this conference come from United Kingdom, Norway, Cyprus, Japan, Malaysia, Iraq, India, Singapore, and Indonesia. Several excellent keynote speakers and will present in this conference. Our outstanding keynote speakers consist of Professor Zhong Wei Guang from Uni Emirat Arab, Professor Andri, Professor Widi, and several invited speakers. Ladies and gentlemen, I would like to express gratitude to our partner university from UITM, Hiroshima University, Japan, ITB, ITK, and sponsored by PT Hutama Karya, PT Pembangunan Indonesia, and PT Nindya Karya for all the generous support in this event. I would like to express sincere thanks to Rector of Mula Rector of Bina Nusantara for very kindly delivering welcome remark on this 8th ICST 2023. Also, I would like to sincere thanks to all committee members, especially organizing committee, for the tremendous teamwork effort making this event in reality. My sincere gratitude also goes to Rector of Universitas Mataram, Head of Research Institute Community Services, Prof. Sukartono, for the continuous support throughout the preparation of this successful event. Finally, thank you very much for all persons participants. You are all very important part on our successful in this conference. Becoming close to this report speech, I would like to invite Rector of Mataram University, Professor Instu Bambang Harikusumo, for delivering opening remark to open this conference officially. All in one, I sincerely hope you all enjoy today's conference and thank you for participation. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you so much, Professor Buan Anshari, for the report.
Ladies and gentlemen, let us listen to the first welcoming remarks from the Rector of Venus University, Dr. Nelly Escom MM CSCA, with a round of applause.
Assalamualaikum warahmatullahi wabarakatuh. Good morning everybody, selamat pagi, salam sejahtera untuk kita semua. Honorable Rector of Mataram University, Rector of Bina Nusantara University, Executive Board of Mataram University, Vice Rector, Dean Director of Research and Community Services of Mataram University, distinguished guests and all keynote speakers and all participants of the International Conference on Science and Technology, ICST. Excellencies, ladies and gentlemen, on behalf of Mulawarman University, I am the Rector of Mulawarman University, would like to express our gratitude, thanks and high appreciation to Mataram University that hosted the International Conference on Science and Technology ICST conducted in Mataram University using hybrid platform. We also would like to warmly express our honor to Bina Nusantara University as the host partner in this conference occasion. Mulawarman University really appreciate that the three universities, one from Java Island, one from Lombok Island, and one from Kalimantan Island, could join and collaborate together to conduct this scientific meeting and conference as implementation of the mutual benefit collaboration among us. We also very welcome for the next and future program for other kind of academic and research collaboration. Excellencies, ladies and gentlemen, university has proven to be successful agent for transformation of human life to a better and prestigious level of life toward and happiness, well-being, and prosperity. University is always generating and producing science, technology, and innovation that significantly contribute the transformation of human life and its environment. Therefore, this international conference that take the theme sustainability in research and science and technology for a better life is very strategic to answer the future need for human life and higher education development. We do hope that the conference will give opportunity for researchers, scientists, and also other experts to warmly discuss and sharing knowledge and new trending topic of science and technology that will significantly contribute for the advance and development of science and technology. We also hope this occasion will strengthen the collaboration among us for further program in the future after this conference. As additional information, Mulawarman University has indeed a very prospective future. Since Indonesian government has decided to move the capital of the country to East Kalimantan, where Mulawarman University is located. We intend to look for and invite the prospective and potential national and international partners to work together in supporting our new capital city with the prospective research and academic collaboration, especially in the topic of Nusantara Capital City Development. Finally, we do hope that the International Conference on Science and Technology would result in the fruitful outcomes such as articles and proceedings published in highly reputable journal. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh. Rektor Mulawarman University, Profesor Dr. Insinyur Haji Abdul Nur MSI APU.
Ladies and gentlemen, we apologize for the mistake and thank you so much for the Rector of Mulawarman University, Professor Dr. Insinyur Haji Abdul Nur MSI. Now, ladies and gentlemen, we will listen to the next welcoming remarks from the Rector of BINUS University, Dr. Nelly Eskom MMCSJA. Let's welcome her with a round of applause.
Distinguished Professor, keynote speakers, ladies and gentlemen. Yang terhormat, Profesor Insinyur Bambang Harikusumo, MRSD PhD, Rektor Universitas Mataram, Profesor Dr. Renat Agus Rubianto, MMSC, Rektor Institut Teknologi Kalimantan, Dr. Insinyur Haji Abdul Nur MSI, Rektor Universitas Mulawarman, Profesor Dr. Insinyur Siti Hiliana MSI, Vice Rector of Academic Universitas Mataram, Profesor Akmaluddin ST MSc Eng PhD, Vice Rector of Planning, Collaboration, and Information Systems, Universitas Mataram, Profesor Dr. Insinyur Soekartono, MR, CP, PhD, from Research and Community Service Office, Universitas Mataram, Profesor Dr. Nasmi Herlina Sari, ST, Head of Quality Assurance and Academic Development, Universitas Mataram, Profesor Insinyur Muhammad Sarjan, Director of Postgraduate Program, Universitas Mataram, Profesor Buan Ansari, ST, MSc, and PhD, as a Chairman of ICST. Good morning. Thank you very much for coming today. It is my honor on behalf of BINUS University to extend a International Conference on Science and Technology, ICST 2020, which the theme is Sustainability in Research on Science and Technology for a Better Future, which underscore the shared commitment to address global challenges, particularly in the development of research and technology. And BINUS University is very glad to be a co-host of this conference. In ICST 2023 is a platform for researchers, scientists, and professionals from various disciplines and backgrounds to exchange ideas and collaborate. We believe that this conference will ignite, inspire all of you to actively participate and share, because through this interaction, we will collectively enhance research quality and contribute to enriching science and technology, which will definitely benefit and impact for the society. I also would like to salute everyone who has contributed to this success of ICST 2023, including the organizing committee, participants, experience. Let's work together to build a better and more sustainable future. Thank you for being a part.
Thank you so much, Dr. Nelly. The next is the opening remarks from Rector of University of Mataram,
2023.
Professor Insinyur Bambang
Enjoy the
Harikusumo,
conference.
MAger, ST, PhD. Oh, let us welcome him with a round of applause.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning. And also the chairman of ICST. The Honorable keynote speakers, Professor Andri Dianugraha, Professor Widi Sunario, Professor Mikhail Y. Roleda. He's from the Philippines, and Professor Jung Wei Guan, Professor Muhammad Asrimat Peridi, and last but not least, Professor Tergius Gautianus. And also the honorable invited speakers that I cannot mention one by one because it's difficult for me to read one by one. But thank you for coming. Thank you for sharing your skill, knowledge, and experiences. Conference on the 8th International Conference on Science and Technology 2023 with its theme, the sustainability in research on science and technology for a better future. I'm honored to stand here before you as Director of University of Mataram to open this and welcome you all to this conference that is located in the very beautiful location in Hotel Raja Kuta Lombok. And all you who attend directly this conference here, I hope that you will have time to enjoy the beauty of nature around this villa. But most importantly, it is the choices we made will profoundly affect the quality of for the generation to come. It is essential that we harness the power of science and technology to address the pressing issues of our time. And this conference provides the platform for the crucial dialogue. Through this opportunity, we will engage with experts in various fields, share your research, exchange ideas, explore new perspectives, and perhaps build collaboration in the near future. Because this conference is not just about academic presentations, but also it is about forging connections and building partnerships. Together, we can tackle the challenge that lie ahead. We can explore sustainable solutions that encompass not only environmental issues, but also the economic and social dimensions of sustainability. Our discussion can lead to breakthroughs in technology, and our research can inform policy and drive positive change. Ladies and gentlemen, last but not least, let us also give a round of applause to the organizing committee, sponsors, volunteers who have worked tirelessly to make this conference in reality. Your dedication and hard work have paved the way for this exceptional conference. I wish you all a very productive and inspiring conference, and let's the conference officially begins. For you who attend this conference directly here in this location, please spend your time to enjoy the very beautiful beach, beautiful scenery, and try also to see directly the circuit of Mandalika. It's very famous today, and some people said that even though you have come to Lombok Island, but before you see directly the circuit of Mandalika, no one believe that you already stand your foot here in this beautiful island. Enjoy the conference and also enjoy the food. Try to travel around here and probably around this beautiful Lombok Island. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Om Swastiastu, Namo Buddhaya, salam kebajikan, Rahayu. And may God bless you all.
Thank you so much, Professor Bambang Harikusumo.
Ladies and gentlemen, next agenda is prayer that will be led by Mr. Andre Rahmat Kabra, MSE. So to Mr. Andre Rahmat Kabra, stage is yours.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, before we start our activity, let me pray to Almighty God. Let me pray for the smoothness, kindness, and every good things today. I asking permission to all of you to lead the praying session in Islamic way. A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Qul huwallahu ahad. Allahus samad. Lam yalid wa lam yulad wa lam yakul lahu kufuwan ahad. Allahumma salli wa sallim ala nabiyyina Muhammad. Asbahna wa asbahal mulku lillah walhamdulillah. La ilaha illallah wahdahu la syarika lah. Lahul mulku wa lahul hamdu wa huwa ala kulli syai'in qadir. Rabbi as'aluka khaira ma fi hadzal yaumi min syarri ma fi hadzal yaumi wa syarri ma ba'dahu. Rabbi a'udzu bika minal kasal وسوء الكبر ربي أعوذ بك من عذاب في النار وعذاب في القبر اللهم أنت ربي لا إله إلا أنت خلقتني وأنا عبدك وأنا على عهدك ووعدك ما استطعت أعوذ بك من شر ما صنعت أبوء لك بنعمتك علي وأبوء بذنبي فاغفر لي فإنه لا يغفر الذنوب إلا أنت اللهم إني ظلمت نفسي ظلما كثيرا ولا يغفر الذنوب إلا أنت فاغفر لي مغفرة من عندك وارحمني إنك أنت الغفور الرحيم اللهم إني أسألك علما نافعا ورزقا طيبا وعملا متقبلا ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار سبحان ربك رب العزة عما يصفون وسلام على المرسلين والحمد لله رب العالمين. May Allah subhanahu wa ta'ala forgive our sins and bless our life. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Mr. Andrew Rahmat Skabra, MSE. What's next we have, Almer?
Ladies and gentlemen, are you still excited for the next agenda? Because we are about to watch the traditional dance performance from Sanggar Tari Rahayu Lombok Tengah. They will perform Kembang Sembah Dance, which means to welcoming the guests. So let us welcome Sanggar Tari Rahayu Loteng with huge of applause.
What amazing performance.
Thank you so much. Sanggar Tari Rahayu.
Ladies and gentlemen, we will have a coffee break session for 10 minutes. But before that, we will hold a photo session first. So I would like to request everyone in this room to please gather in front of the stage for the photo session. Please. One more time, I would like to request everyone in this room to please gather in front of the stage to take photo. Thank you. Kan Bapak dan Ibu mungkin dapat bergabung bersama kami di depan stage untuk kita ambil foto bersama. Oh ya, Pak Rek. Oke. Mari silakan Bapak dan Ibu. Tidak. Silakan Bapak dan Ibu. Mungkin kalau misalnya sudah full di bawah bisa ke atas Bapak dan Ibu. Yes. Please go to the stage if it's already full in front of me. Ya, sebagian di atas. Alright, I will lead the photo session. Now we will do a formal pose first. Yeah.
All right.
We will do a formal pose first, and I will do a countdown from 3 to 1. Okay, ready, everyone? Okay, 1, 2, 3. Oh yeah, the Secretary of ICST 2023. All right, we'll do one more formal pose. 1, 2, 3. Pose. Do we have any special pose for ICST 2023? Yes, if no. Okay, we will do this, which means lombok. Okay, let's do 1, 2, 3. Pose. And also maybe this pose. Yeah. When I say Unram, you will say Jaya. All right. Ready, camera? Okay, 1, 2, 3. Unram. Unram. Thank you so much. Please come back to your seat. Okay, now what we will have, Almer, after this?
Ladies and gentlemen, before we continue to the plenary session, we would like to have 10 minutes coffee breaks. So,
Oh, all right. Okay. So I would like to invite the committees of ICST also to please join us in front of the stage to take photo together. And I would like to because the coffee break station is already here, and we would like to continue the conference in 10 minutes. So please, you can go outside and check the coffee break and take food and also coffee as much as you want. Thank you. And I would like to invite once again to all of the committees to please join us in front of the stage to take photo.
baik Bapak Ibu Panitia, dipersilakan untuk maju ke depan untuk berfoto.
Now another pose, maybe? We can do another pose. It's too formal. All right, let's do Unra. Thank you. Thank you. Ya, kita Bapak dan Ibu akan memiliki break session selama 10 menit. Silakan untuk dapat mengambil coffee break. Mungkin yang sudah disediakan panitia di luar. Dan kita akan kembali lagi pada pukul 10.10.
Ladies and gentlemen, before we continue to the plenary session, we would like to have 10 minutes coffee break. So enjoy your coffee break and we will see you again at 10 a.m. in the same room. Thank you.
Demi saya Pak petani. Biasa.
Cuman sing akeh itu ya.
Kami nopo boten nopo
Ya. Yang jelas mingkono tengger mangan nasi padang.
kan santen-santenan gitu.
Warungnya ini ya ono jenenge nanti senor Pak ada. Ada mam dicatat gitu. Jadi piring di rape
Dan juga kiron sini.
Iya.
professor in photobiology. He has more than 100 Scopus documents and over 4,000 document citations. And also recently, Prof. Roleda was listed as the top 2% scientists, Stanford University version this year, 2023. So first we will hear the talks from all the honorable keynote speakers, and the time is around 30 minutes, Prof. And after that, we will have a discussion. So first, I would like to welcome Prof. Doctor of Science, Andri Dianugraha. The time is yours.
Okay, thank you very much. So, okay, thank you. First of all, good morning. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi. Let me introduce myself. My name is Andri Dianugraha. Actually, I'm from Institut Teknologi Bandung, West Java, Indonesia. So this is the title. I'm going to talk about seismic structure around Lombok, Kalimantan, and Sulawesi region, Indonesia, derived from tomographic inversion. So there are our teams from ITB, BMKG, Geological Agency of Indonesia, University of Cambridge, and NTU, and also Australian National University. Actually, I'm from Geophysical Engineering Department. So basically, I'm seismologist. We use seismic wave that generated by the earthquake that recorded by seismograph station on the surface, and we try to see the underneath or the subsurface or the interior of the earth by using a seismic wave. Okay. So actually, I would like to talk several studies that we have conducted. For example, travel time tomography, and also at seismic attenuation tomography, and also immunoid tomography in the several regions in Indonesia, including that I said before, Lombok, Kalimantan, and Sulawesi region. Please to the next slide. Please next slide. This is for the first. I would like to talk about the field survey in Lombok 2018. Actually, in 2018, on July and August, there were a strong earthquake occur in the northern part of Lombok Island. So at that time, we from ICB and also from EOS, BMKG, and PPMG deploy some temporary seismograph in the northern part of Lombok to see, to detect, to record the aftershock event, and also to see the seismic structure underneath. And next, actually, the Indonesian region is affected by 4 major tectonic plates in the world, including Indo-Australian plate in the southern part of Indonesian region, Eurasia, and also Pacific plate in the east, and Philippine Sea plate in the north. As consequences, of course, there are many earthquake event, and also we have also active volcanoes actually from west to the east of Indonesia region. Our government published National Seismic Hazard of Indonesia. Actually, this is the new one version published in 2017. Actually, this is the Lombok Island. Lombok Island itself, for the geological setting and tectonic setting, is affected by, for example, in the southern part, we have Indo-Australian plate subduct beneath the Lombok Island, and atlas that we call forest bake atlas subduct beneath the Lombok Island. And then the next slide. This is the main event or the strong event earthquake that occurred in 2018 in the northern part of Lombok, starting from July 29 with the magnitude 6.4, and then August 5 with the magnitude of around 7, and also on 19 August with magnitude 6.3 and 6.9 at that time. Soon after the first event, we are from ITB visited the northern part of Lombok and deploy the seismograph, temporary seismograph at the time, collaborate with the BMKG and NTU Singapore, and we deploy around 13 to 21 temporary seismograph at the time. And this is the epicenter distribution of the aftershock of the earthquake sequences in Lombok 2018. Next. Okay, sorry, next slide. This is the situation during the seismograph deployment around the source region of the 2018 Lombok. Actually, for example, this is the reverse blue triangle, stand for the temporary seismograph at that time. We deploy and we record the aftershock event for about 1 month, starting from August 3rd up to September 9, 2018. The number of stations from 13 to 21, and this is the situation in the field. That this is photo from Zulfakrija at that time. So next, this is the first step. We identify the recording of the earthquake event on the seismograph. For example, this is example for the one event recorded by several temporary seismograph, and then we see and we pick for the P-wave arrival time and S-wave arrival time as input for the hypocenter determination. And next, and actually the next step is hypocenter determination, and we can see here actually the seismicity concentrate in the northern part of the Lombok and is clustered into region around 20 to 40 kilometers across here. And actually, this presumably represent the seismogenic segment length for this section of the Flores Bake Actras, or that we call FBT. And also we can see here, this is along the longitude, this is depth, and this is along the latitude, and this is depth. In depth, most seismicity is located shallower than 20 kilometers, which is the depth of the foreman shock captured by the local network. Next. And then we can see here, so actually the temporal and spatial clustering of the seismicity, we suggest that the Flores oceanic crust in the north, to the north of Lombok, is segmented. For example, this is the segment 1, and this is the segment 2, and this is the segment 3. And also this is the vertical cross section. This is the initial location. This is the relocated location from south to the north. And we can see clearly here, actually, the large earthquake to be generated by movement between the Flores oceanic crust, subducting plates, and operating island of Lombok. And the event actually related to the subduction system of the Flores oceanic crust beneath Lombok with a backup thrusting mechanism. This is only for 1 month. So many aftershock event at that time. Next. And then we use the earthquake data, especially for the P and S arrival time, to determine the seismic velocity wave. For example, for P wave velocity or PV, and shear wave velocity or VS, and also PP over VS ratio around the 2018 Lombok earthquake region. And actually, we use 20 seismic stations that that indicated by a reverse triangle here. And also this is the aftershock event that we use for the input for the travel time tomographic inversion. Okay, the next. And this is the result of the tomographic inversion result. The first column for the PV structure, the second column for the VS structure, and the third column for the PP over VS ratio. Blue and red color stand for the high and low velocity anomalies relative to the 1D initial velocity model. And then there are actually some region that we interpret. For example, this is region 1. Maybe it is not clear here. This is R1, for example, for PP structure low, and for PS structure low, and for the ratio PP over VS moderate to high value. Actually, this is extend from west to the east along the North Lombok coast and coincide with the aftershock cluster, which define the rupture fault, shortward diving, and parallel to the Flores backup thrust. And the other feature also, for example, this is R2. This is for VV, VS, and VP over VS structure, respectively. We show something like low VP, low VS, and high VP over VS is further to the south beneath Rinjani volcanic complex. Actually, we observe a little seismicity and gently dip to the southeast. Next. And the other feature, for example, R3, yeah, maybe around here, sorry, I couldn't see clearly here. R3, R4, and R5. Next. Actually, this is the final model for the tomographic inversion result. And actually, this is the PP structure from regional tomography from previous study. Show the subducted Indo-Australia plate beneath Lombok and south diving lithosphere directly north of Lombok. And we can see here, this is Indo-Australian slab as indicated by blue color. And this is the Flores oceanic crust in the north, as is also indicated by blue color here. And then sorry. And this is the 2D interpretation, yeah, for the crust beneath northern Lombok. For example, this is based on PP structure along the section from the south to the north. Yeah, actually, this is Flores oceanic crust represented by a gray block that subducted southward and regurgitate weak zone located in the middle of this block, as indicated by red color here. The presence of the weak zone probably the result of the pervasive fracturing and presence of the hydrous fluid from above and below Flores oceanic crust. And this is the final model of 3D cartoon from our travel time tomographic inversion result. Clearly, this is north and this is south, and this is the Lombok Island. So 3D cartoon of southward subducted Flores oceanic crust represented by blue block. Yeah, it's clearly. Actually, this is nice model, and red color zone stand for the weak zone, as indicated by R1 or region 1 in every single node. So the most of the aftershock core of this zone. So this is the Flores oceanic crust. Next. And then we extend our study by seismic attenuation tomography. Actually, same to see the seismic structure underneath, yeah, to determine 3D seismic attenuation structure that we call QP and QS around the aftershock source region of the 2018 Lombok earthquake in Indonesia. Next. Okay, actually for the seismic attenuation tomography, we need the T-star or operator attenuation as input for the attenuation seismic tomography. And then we have to determine the T-star and also that we call low-level spectra omega 0 and corner frequency by using spectral fitting equation, something like this. And then we selected the data consists of 914 earthquake event, result in 6,200 around piece wave T-star for the attenuation tomographic inversion that we can see something like this. And that, next slide, sorry. Sorry, the next slide. Next, actually this is the equation for the attenuation tomographic, the relation between attenuation, Q model, velocity model, and also ray path segment length along the medium. For the inversion, we started from the QP was 300, and the initial 3D PP from the travel time tomography of our previous result. Next. And this is the result of the tomographic attenuation. This is vertical cross-section. Actually, from north to the south, this is for PP, this is PP over PS ratio, and this is for the QP, or one for attenuation. And the next, this is the final model for the attenuation seismic structure. And this is PP structure. We compare, we can see here the 2D interpretation of the crust beneath Lombok based on a QP model along the south to the north direction. And we can see here, this is the schematic diagram, yeah, around the Lombok Island. This is the north, this is south, this is Rinjani volcano. And we can see here in inverted triangle, make the surface fault location. And the yellow star here, denote for the M magnitude 7 that occur on August 5, 2018, which has a thrusting fault mechanism. And also the white dot indicate the location of hypocenter with the radius of 3 kilometers from the center line. The red line indicate the fault from the previous study, and the black line indicate the fault from also previous study. And the green area, for example, around here, near the red line fault indicate the high temperature fluid content, and the dark blue sediment here represent the upper Miocene limestone, and the green sediment layer represent the Pliocene to present young sediment, and the light blue layer represent the ocean. Next. And this is the ongoing study. Actually, we have been conducting seismic velocity structure in Borneo, yeah, including Kalimantan, and also Makassar Strait, and also Sulawesi region. And this is from our PhD student, the ongoing research. Actually, the result of the study is still under review. And we use the number of seismograph around 1,108, including 75 stations from BMKG, Meteorological, Climatological, and Geophysical Agency of Indonesia, and 4 stations from the Malaysia. And we also deploy temporary seismograph around region and Kalimantan, cooperation with the University of Cambridge, around 29 stations. So this is the period of data that we use, around 8 months, from October 1st up to June 1st, several years ago. Next. And this is the result, the zero velocity structure, or VS, derived from ambient seismic noise tomography around Kalimantan, Makassar Strait, and Sulawesi. This is at depth of 8 kilometers, depth of 14 kilometers, depth of 30 kilometer, and a depth of 40 kilometer. Blue and red indicate high and low VS value. We can see here, we can observe some, for example, this is low VS in the Kutai Basin at depth 8 kilometer and 14 kilometer, maybe related to the something like sediment, thick sediment layer in the region. Actually, next. Actually, that's all that we had. Actually, thank you very much for inviting me to come here. It is a great pleasure for me to come here. Thank you very much. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Prof. Andri, for your splendid talk. So please stay here until we finish the other keynote speakers, and then we will do discussion later on after all the keynote speakers have finished their talk. So I would like to welcome the second keynote speaker is Prof. Widi Sunario, PhD from Mulawarman University, and he is attending on site. Then welcome, Prof. Widi. So Prof. Widi has over 100 citation scopus documents and currently in the field of agronomy, plant biotechnology, and plant molecular biology. He is currently the professor for biotechnology plant at the Faculty of Agriculture, Mulawarman University. So Prof. Widi, you're welcome. The time is yours.
Saya mungkin ke bawah aja ya. Di bawah aja. Biar ke bawah aja. Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, thank you very much for the opportunity. For me, my name is Widi Sunario from Mulawarman University. I would like to present one of our projects. This is about very famous fruit from Kalimantan called as lai durian, one of the durian species. Please. Okay, the title of my presentation is Lai Durian, a local super rare durian from East Kalimantan, genetic and physiological study for fruit improvement. Next. Indonesia is the second mega biodiversity of the Brazil, including in durian species. And from a total 20 durian species in the world, 18 species is existing in Kalimantan Islands. And this makes that Kalimantan known as the center of durian species. Next. Due to the pollinated, open pollinated characteristic, the genetic diversity of durian species is enlarging and spreading significantly. And durian species is a tropical fruit that can at a very broad range of climate and season. We have some example of Durio species in Kalimantan Island, Durio dulcis or lahong, Durio oxalenus, or local people call as kerantungan, Durio testudinarum, Durio kura-kura. Next. And Durio zibotenus, all of us know about this, durian. Durio cutejensis, we call as a lai or pempekan in South Kalimantan. Durio excelsus or apun. Next. Durio glandivorus or sukang. Durio glaviolens, red. Durio graviolens, yellow. Next. All of these Durio species existing in Kalimantan Island. From the study of the nutritional value of the Durio species, we know that durian fruit has a very good nutrition for human body, especially for the energy, sugar, carbohydrates, and also minerals. Next. Next. And in Kalimantan, we have a national superior variety of durian, around 14 variety, durian liquid, durian salisun, matahari. All of this national superior variety come from Durio zibetenus, Durio cutejensis, and also the crossing species from Durio zibetenus and Durio cutejensis. Next. And one of them, Durio cutejensis and Durio zibetenus has different, significant different characters. As if we look at the Durio zibetenus, has character as sweet, subodors, wet and soft texture, white, yellowish, yellow and yellow goldish, and sometimes contain high alcohol and also high water content. But for the Durio cutejensis, it's sweet, odorless, dry and dense texture, and the color is very interesting from yellow. Next. Next. And one of the national superior variety from East Kalimantan that I would like to present here is Lai durian. This is a very interesting variety because has very remarkable properties and superiority, like sweet, odorless, cold yellowish, and so on. That's why this Lai durian many times win the competition of durian in East Kalimantan. But until now, the taxonomy or the identity of this fruit still unclear. Sometimes identified as a Durio excelsus, and other researcher call as a Durio pronatus. That's why in this occasion, I would like to present about the study of this taxonomy status. Next. And this is the variation of Lai durian from East Kalimantan. Next. And my projects focus on the study of the taxonomy status, the exploration of the origin, also the study of diversity and distribution of the Lai durian. Also, we would like to improve the economic value, and also, of course, the development of the cultivation technology. Next. The first experiment is about identification of the taxonomy using morphological character. Next. We have explored durian from all area in East Kalimantan. Next, including and we found durian in Kutai Barat, in Penajam Pasir Utara, in Balikpapan, Samarinda, Kutai Timur, Kutai Kartanegara, Berau, Nunukan. Almost all district in East Kalimantan we can found durian. Next. Next. And this is a 7 durian found during our exploration in 2015 until 2017. Next. After we conduct a morphology study, we find that light durian has a morphological character intermediate between Dureocybetenus and Dureocutegensis from the flower colors, from the fruit, from the aril color, from the seed, and also next from the leaf characters. We can see that light durian so intermediate between Dureocybetenus and Dureocutegensis. Next. And from the measurement of the all morphological character, we can also indicate that light durian has intermediate character between Dureocybetenus and Dureocutegensis. Next. Next. And from the nutritional value, we can also suggest that light durian has Dureocybetenus and Dureocutegensis. Next. And from the study, we can conclude that based on the plant morphology, fruit performance, and nutritional value, there is strong indication that light durian is originated from the natural crossing between Dureocybetenus and Dureocutegensis. Next. The next experiment is to study based on the genetic So we use DNA barcoding to clarify the finding from morphological study to make sure that light durian is the natural crossing between Dureocybetenus and Dureocutegensis. Next. And what is the DNA barcode? I think most of us know DNA barcoding, especially who work with molecular marker and also molecular analysis. DNA barcode is a short statement for biology question, and we can use for the plants. Usually, we use Ripulosa biphosphat carboxylase large unit or LPPM to identify the species. Next. Using this DNA barcoding, we can do first sampling. Then usually we use a leaf or a cambium for DNA extraction, and then amplification using RPCL primer, and then amplifications, and then afterward we do DNA sequencing and then data analysis. Next. And this is the example of DNA amplification using RPCL primer. Next. And then this is the result of amplification using RPCL for around 14 variety of Durian species from East Kalimantan. We can look at here a band, specific band with size 530 base pair. This is very specific for RPCL primer. It means that all of variety can produce amplicon in the same size. Next. And we do sequencing. And after we get sequence, we have to analyze to make a alignment, to make a same size. And then we do some analysis like cluster way to make a difference analysis. Next. And from the difference analysis, we can look at the distance among varieties. It's around 0.018, and the closest genetic distance is between Holei Sentawar and Durian Montong, and the farthest distance is between Lai Mandong and Durian Rinbut. And then we make a phylogenetic tree. Next. And from the phylogenetic tree, we can see very clear here that Durian Kani and Durian solution very close related. Durian Montong and Holei Sentawar also very close related. And Lai Mandong, Lai Batua, Lai Kayan also very close related. It means that we can see here very interesting finding that Durian Montong and Holei Sentawar very have a relationship is very close. Durian Montong is Durio zibetinus. The other one, Holei Sentawar, is Lai Durian. It means that because of RBCL derived from chloroplast genome, that's mother derived from the mother chloroplast. It means that the mother of Holei Sentawar is Durian Montong. This is very very interesting finding from this here. So we can know from the analysis that the paternal origin of the Holei Sentawar, one of the very important Lai durian from East Kalimantan, is from Durian Montong. It means that maybe in some time ago, there is a crossing, artificial or natural, between Durian Montong and also Durio cutejensis, and produce the Holei Sentawar. Next. And this is the analysis of the paternal study that I already mentioned, that Lai durian, especially Holei Sentawar, originated from the natural crossing between Durian Montong and also any Lai or Durio cutejensis. Next. From this study, from this genetic study, we can make a conclusion that Durio sp is open pollinated cross pollinated plants, and if we want to improve, the Lai durian as a commercial variety, for the next development, we need good or superior plant material. That's why we also conducted a study mass propagation of the plant material, because Durio species is open pollinated or cross pollinated plants. We need a clonal propagation to ensure the genetic similarity of the plant material. And then, because of the limited seed as a rootstock for the clonal propagation, we try to use Durio zibetinus or Durio cutejensis as a plant rootstocks. Next. This is the experiment to use rootstock from Durio zibetinus, Durio cutejensis, and also from the light durian, using two kind of grafting, using chip budding and also using cleft grafting. The objective of this experiment is to find the effective way for mass vegetative propagation of light durian, and also we look for alternative the effective rootstock between Durio zibetinus or Durio cutejensis. Next. And from the experiment, we can see here that using chip budding resulted in the highest percentage of successful grafting compared to the cleft grafting. It means that we can choose chip budding for the mass propagation of light durian. Next. And from the observation of plant growth, we can see here also that using the most effective grafting is if we use rootstock from light durian and also the cyan left durian, but we can also use Durio zibetinus and Durio cutejensis also successful to make a grafting from Durio zibetinus and Durio cutejensis as a rootstock. But the highest effectiveness is from if we use the rootstock and the cyan from Durio from the light durian. Next. This is the development of plant growth of the grafting. Next. Next. And from this study, we can make a conclusion that the chip budding had a higher percentage of successful grafting compared to the cleft grafting. And for the interspecific grafting between Durio zibetinus and Durio cutejensis, we can use Durio zibetinus and or Durio cutejensis as a rootstock also. Next. Still have time? Yeah, okay. Next. And then I think this is the last experiment that I would like to present. We also want to increase the productivity of the light durian Kalimantan. Problem to increase the productivity of light durian is in Kalimantan is the plant growth of light durian really depend on the area or microclimate conditions. The second also really depend on the season. For the flowering initiation and fruit formation. That's why we conduct and we try to do off-season initiation of flowering. It means that usually in East Kalimantan, the flowering time of the light durian in March until March, April, and also uh, June and July, 2 times a year. But we would like to conduct in out of this season. And from the literature, we know that the flowering initiations controlled by the GA pathway and metabolism in the plants. That's why we in this experiment also we use gibberellin as one of a treatment to initiate flowering of season. And we use also some other retardant and also plant growth regulator like paclobutrazole and prohexadione CA, and also some auxins. And from other experiment, there is also information that if we use strangulations, also can trigger the flowering initiation. Next, this is the treatment for our experiment. For the strangulation, we use 2 mm iron wire coiled to the stem 3 times for 1 month. And the other for paclobutrazole, we apply by flushing to the soil surrounding the plants. And for prohexadione and gibberellin, we spray the liquid of this chemical treatment to the plants. Next. And this is the fertilization of the plant before the treatment, because we need to ensure that the plants have sufficient nutrients for their growth and also for the reproductive time. Next. This is the also fertilization using inorganic fertilizer. Next. And this is strangulation using iron wire for months. Next. This is the spraying of paclobutrazole and gibberellin. Next. The result is that using paclobutrazole, prohexadione CA, and gibberellin can induce off-season flowering initiation, but strangulation doesn't induce the flowering initiation. And the highest result of the flowering initiation is from prohexadione GA prohexadione calcium treatment. Next. And this is the picture of a flower of light durian. The color, the specific color of light durian is pink. If Duriocutegenesis of light is red, red, maroon red. For the Duriocutegenesis is white or yellowish white. Yeah. Next. And this is the fruit set formation Next. This is also process formation. Next. This is the developing fruit, like durian. Next. And the conclusion is that all of season flowering treatment could trigger like durian flowering and fruit set formation solely except strangulations. And CA prohexadione treatment could produce the highest number of harvested fruit per plant. It means that off-season initiation flowering in durian can be conducted successfully in this study. Next. And for the in progress works, now we conduct cytogenetic study to look at possibility of the ampidiploid hybrid chromosomes, and also we do genetic diversity study using IS as marker. It's already done, but still in data analysis. Next. Thank you very much for all people that contribute for this study. Profesor Dr. Nurhasana, Profesor Rusdiansa, Bapak Rahman, Dr. Medi Hendra, Dr. Rudarmono, Dr. Hadi Suprato, Dr. Hadi Pranoto, Dr. Sadarudin, and also student in my lab, Lucianto, Krishna, Aris, Ahmad Mulyadi, Puput, Riza, and Sarful. We also make a collaboration with UPTD PSP TPH Provinsi Kalimantan Timur and also Naresuan University Thailand. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Prof. Willy. So you're welcome to stay here for the discussion session. But we have one more speaker. He is joining us online via Zoom, Professor Dr. Renat Michael U. Roleda. So I sometimes call him Prof. Mikey. Prof. Mikey, are you with us now? Not yet. So Prof. Michael U. Roleda is a professor from the University of Philippines. He graduated his PhD from the University of Bremen, Germany, in the fields of psychology and also plant biotechnology. Yeah, and recently, Prof. Michael U. Roleda was also listed as the top 2% scientist in the world, version from the Stanford University this year in 2023. So I would like to congratulate on that also, Prof. Mikey, for making the 2% list.
Thank you for that, Doc
Are you with
Eka.
us?
Yes, could you
Okay.
hear me?
Yeah, I can hear you well. So time is yours from now, Prof. Mikey. You have 30 minutes, but if you want to use it less, it's up to you. So okay, the time is yours. Please start.
Okay, thank you very much, Doc Eka. So, colleagues, administrators, conference organizers, and the secretariat, ladies and gentlemen, good morning. First of all, I would like to thank the University of Mataram for inviting me to deliver a keynote speech. I hope this can further existing collaboration to address the pressing problems that the tropical seaweed industry is experiencing. I actually have a current collaboration with Doc Eka, and I hope to find more collaborators from Indonesia. We all know that Indonesia is the primary, that is the #1 producer of carrageenan seaweed biomass in the world. But the crop production has been declining in recent years across all tropical seaweed farming countries. So some of the reasons are climate change, for example, ocean warming, and then the prevalence of pests and diseases, and the declining seed stock quality, thus affecting seaweed productivity and biomass production. So I hope this presentation will be useful for our colleagues doing seaweed research to address these challenges, and also will be appreciated by the general public. I know this conference is very has diverse expertise to appreciate this talk, because you might not know this, that our daily lives is basically supported by the seaweed-derived polysaccharide. I think most of you, or maybe some of you, know the different uses of seaweed. So, for example, a number of coastal inhabitants, especially women, so they are engaged in seaweed farming, which provide additional family income to support the education of their children and others. So the raw seaweed that is processed into the semi-refined carrageenan or the refined carrageenan has a lot of uses. So they are mainly used for food applications. So what we are eating, most of it has carrageenan. And maybe you also know that our toothpaste that we use every day in the morning, they have carrageenan. So they are also used in medical, pharmaceutical, and cosmetic applications, among others. So what are eucheumatoids? So maybe first to look at it taxonomically. So eucheumatoids are red seaweed. So they belong to the phylum Rhodophyta, class Floridophyceae, order Gigartinales, and family Solidaceae. So they are morphologically plastic. Initially, all of this carrageenan producing seaweed, they used to be classified under one genus, which is the Eucheuma. And then recent advances in taxonomy, there have been some reclassification. So what used to be Eucheuma cottoni now become Cataphycus alvarezii, and what used to be Eucheuma spinosum now become Eucheuma denticulatum. In the right-hand side, you could see here different morphology of this taxa. And from the morphology here, you might think that they are different species, but after doing DNA barcoding, all of these four different individuals turned out to be Cataphycus alvarezii, which exhibit these different morphologies. Okay, so the primary taxa under the general classification of eucheumatoids are Eucheuma, Betaphicus, and Cataphycus. So Eucheuma produces iota carrageenan, Betaphicus produces a combination of beta carrageenan with some kappa carrageenan, and Cataphycus produces kappa carrageenan. And some minor taxa include Eucheumatopsis, Mimica, and Cataphycopsis. So to appreciate the importance of our work on seed stock selection, maybe it is important to know the life cycle of eucheumatoids, which has a triphasic, what we call as a triphasic life history. So this means that it is much more complex than maybe the life cycle of human beings. No? So here you could see that they have a haploid and diploid stages. So if we look at the tetrasporophyte first here in this right bottom corner, you will see that the tetrasporophyte produces tetraspores, which germinate and produces male and female gametophytes. So they actually have male and female individuals, which are haploid. And then after fertilization, the carposporophyte form something like a parasite on top of the female gametophyte. And then the carposporophyte produces carpospores, and then shedding of carpospores and then germination will develop into tetrasporophyte. So basically that closes the life cycle of a typical eucheumatoid. So that is as simple as I could let people appreciate on the life cycle, that it is not, it is quite complex, no? And from a seaweed perspective, this is a very complex life cycle. Okay, so the main objective of our initiative is to address the declining productivity and biomass production, primarily in the Philippines. But this initiative is also applicable in other seaweed-producing countries, like, for example, Indonesia. So the goal of this project is to avoid crop failure due to climate change and disease outbreak. So to develop new seed stocks from spores, they should be adaptable to climate change. They should have some kind of resistance to disease, and should also have superior growth performance and carrageenan yield and quality. Is there some kind of... Is everything okay? I was hearing some background noise. Okay, anyway, to continue, if everything is okay. So here is a photo showing the carpospores that is released by the cystocarp, and then the tetraspores that is released by tetrasporophyte. We allow them to germinate on a substrate. Most often, this is a glass slide, and then let them grow into a certain size class. And then after that, we bring them to a bigger container, a circulating aquarium with moderate aeration in an indoor hatchery or indoor nursery. And then when it reaches a certain bigger size, we bring them to the outdoor nursery to grow them. So in collaboration with another partner, C6 Energy, who developed a growth chamber with a recirculating water system. So I hope you see the... Okay, so the media doesn't seem to be working, the video. So this is a recirculating system, which basically try to simulate water movement, which we hope we could promote higher growth rate in these sporelings. And then after reaching a certain size class, we bring them to our aquarium and also some raceway tanks wherein we grow them into a sizable biomass, which we will eventually distribute to our collaborating seaweed farmers. Is my presentation still okay? The slides, are you seeing them? Hello?
Still okay, Brad.
Okay, thank you. So for example, so the media doesn't seem to be working. There's supposed to be a video here. Yeah. Okay. So this is our aquarium setup in our outdoor hatchery. And each of these tanks contain different strain. You could see different colors. So euchumatoids are basically red seaweed, but you could see brown colors, green colors, and dark red colors. So this is an example of our raceway tanks wherein we cultivate specific strain on lines. Okay. Another example showing a close-up photo, different morphologies, different colors, a greenish morphotype, and some dark red. So still some green, red, and you could also see some multicolored strain or cultivar. So these are seedlings that we are growing in our hatchery. And after some time, if we have enough biomass, we share this to our collaborating seaweed farmers. Okay, so like I said, after we have enough biomass for harvesting, and this is either donated to our collaborating seaweed farmers, and at the same time, we measure the growth rate in the hatchery. So this is an example of the 12 different gametophyte strains. These are the strain number, and this is the different tetrasporophyte strain, different strain number, and you could see that the growth rate is strain specific. So the growth rate could vary from a low of 3.37% to as much as 11.56% growth rate, which is very good growth rate under hatchery condition. Okay, so from the biomass that we harvested in collaboration with some Portuguese colleagues, we also submit them for carrageenan extraction to analyze the yield and the viscosity. So in the left-hand side column, you could see here the different strain number, and they use 6 different kind of extraction protocols. So from the study made, you could see that also different strain could also exhibit different carrageenan yield and quality. And we have shown that a specific strain called GN7 has the highest yield and the highest viscosity in a specific extraction protocol. So we could conclude that growth rate and carrageenan yield and quality is strain specific. So it is very important to identify the specific strain that could be given to seaweed farmers for further cultivation. Okay, so we also have our seed bank, and we collect wild samples, and we have different species. So we have 3 different species of Capophycus: Capophycus alvarezii, Capophycus striatus, and Capophycus malaysianus. And we also have Eucuma denticulatum, Capophycopsis cottoni, and Betaphycus species. So in our seed bank, we have a total of 289 eucumatoid strains. And then,
I'm sorry, Prof. Mikey.
Yes.
I have to interrupt you first. Can we pause a little bit because we have a technical problem in on-site connection?
Okay.
So we are waiting for the internet to come back on. Sorry, I will tell you when we have it back on. Please wait a minute.
Should I go back to a certain slide or something, or this? Okay.
Yes, I think the one where you are introducing the wild collection. No,
Okay.
it's here. Yeah.
Okay.
So, Prof. Mikey, because we have more participants joining us online,
Yes.
so we can continue. Please continue your talk. Sorry
Okay.
for the trouble.
Okay, thank you. So I hope on site it is now okay. Okay, so to continue, so I was saying that we are also doing so as mentioned, if I may continue, so we are doing wild collections and we have encountered different species. So we have 3 different species of Capophycus alvarezii, Capophycus striatus, and Capophycus malaysianus. And then we have Eucumat and Ficulatum, Capophycopsis cottoni, and Betaphycus species. So we also encountered Mimica arnoldii, but this is a very difficult species to culture. We tried to bring them in our lab and we were not successful. No? So we maintained this different wild samples in the lab, and then we try also to cultivate them for to propagate them in our hatchery. So among the different strains that we see that is fast growing in the hatchery, in collaboration with different seaweed farmers association, different NGO, or the local government units, we do outplanting of our high-performing strain. So this is an example where in collaboration with local government unit, we outplanted a high-performing strain from our hatchery, brought them in the field, and we also observed very high growth rate. So, and we one of our activities is also to help renew the knowledge of our seaweed farmers. So we do seaweed farmers training and workshop to educate them further on the recent advances and how to address problems related to disease and adaptation to climate change. So we're basically capacitating our seaweed farmers to improve their livelihood. So I think that was the last part of my presentation. So I thank you for your time listening to my talk. And I would like to thank my students, so without which this work will not be possible. So most of this work are the research for the master's and PhD work of my students, as shown in this slide. So we work a lot near the ocean, so we spend some nice time in the mountain. Thank you.
Session of the keynote speakers, I would like to welcome 3 questions first from the audience. Please introduce your name and address the question to the specific keynote speaker.
Thank you very much for giving the time to ask the question. I have to.
Wait, before Prof. Andri, can you come?
Okay. Thank you for giving me the time to ask the question. I have 2 questions. Sorry. The first question for Prof. Andri Dianugraha. It's very interesting presentation, sir. Thank you very much. I'm also interested in earthquake. We have a group in Mataram. We call BIU Geomagnetic. Collaborate with Ms. Teti Jubaida and other colleagues over here. So my question is, in terms of seismic activity before large earthquake, yeah, actually that's occurred in Lombok. is there any correlation between high seismic activity surround the large earthquake before the large earthquake? Is there any correlation seismic activity before the large earthquake? I mean, the day 1 or 2 days before the real earthquake, is there any seismic activity? Uh, I mean, small earthquake to become a large earthquake. I mean, affected the large earthquake. That's for Mr. Fadrian. And then the second question for Prof. Vidi Sunario. I mean, as we know in Lombok Island, we have a lot of variant durian. Yeah. Over here we have kane and etc. etc. In terms of weather and land condition, is there any possibility to grow lai durian in Lombok Island, sir? Thank you very much.
You're welcome to answer, Prof.
Okay, thank you very much, Pak. Okay, thank you very much for the interesting question. I think we can collaborate, Pak, in the future. So actually, for the Lombok earthquake in 2018, for our team, we deploy seismometer after the main shock. So actually, we do not have the foreshock data. Maybe BMKG has. So actually, in the several case, the other large event, maybe several studies also show there is something like foreshock or before the main shock and aftershock. In this time, we just start after the main shock actually. So we do not have data about the foreshock. But maybe for the other cases, there are something like small event before main shock. It could be, but in the in the other studies. So it's very interesting to study about that. Maybe we can see something like a precursor or something like that. Yeah.
In terms of Lombok Island, I think we have a multi-shock, yeah.
Yeah.
Multi-shock. I mean, before the second earthquake in 7.0,
yeah,
and then 5.8, I think. I mean, is there from your
the data. Okay, we will try. Actually, thank you very much, Pak.
Yeah, okay, Pak Prof. Vidi.
Okay, thank you very much for the questions. As I mentioned in the slide, that's Durio species can grow very broad range of climate and conditions. So we can find durian everywhere in Indonesia, in Sumatra Island, Java Island, Kalimantan Island, Sulawesi Island, Bali, Lombok Island. It means that's it can grow in, especially in tropical climate. Like durian, of course, I am sure that can grow also in Lombok Island. But why the Kalimantan is the center of Durio species? Because the very high precipitation. Because you know that durian needs needs a lot of water also, not a lot of water. That's why most of durian plantation and natural durian cultivation in Kalimantan usually grow at the watershed of big river in Kalimantan, like Kayan River, Hakam River, and so on. So because they need also a lot of water. If we short literature in Malaysia, for the example, durian Musang King, in the modern cultivation, they also do like watering, yeah, regular watering to the plants. One that I want to stress is that to initiate flowering, sometimes it need a dry season. For the example, is 1 month. For the example, after this dry season, many durian plants in Kalimantan is flowering now because of dry season. But afterward, need water. This is the condition. So that's why also in Kalimantan, sometimes we can 2 or 3 times durian season. But in Java Island or in Sumatra, maybe 1 or maximum is 2 times. Yeah. Thank you.
Thank you very much, Prof. Anil and Prof. Yudi. Prof. Sarjan, you're welcome to address your questions.
Thank you, moderators. My name is Muhammad Sarjan. I just interested in the topic of the durian from Prof. Widi. I am a planter of durian actually. I have about 20 variants of durian here from Usangking, honey, or local varieties. And also I have a unique variant of the durian, so-called thornless or without thorn or durian gundul. I'm lucky after I've been waiting for 8 years. Finally, I got the durian gundul. So, do what do what your opinion about the durian gundul? Is that a genetically or other cause of the why this can become a gundul? And from my observation, the growth of the fruit is very slow. And this is the real durian. What is your opinion about the durian gundul? Is the unique durian and only one of the world in Lombok. Thank you.
Thank you. Your Prof. You are welcome to answer.
Okay, thank you very much, Prof. Sarjan. It's very great to have a collection 20 variants, local and national variety also. Because Kani is national variety. Also in East Kalimantan we grow also Kani. Toothless durian is just my opinion from the genetic side that this is genetic. This is genetic because you know that sometimes we can find many variant because of mutation. It's just like mutation of the turi. So for the example, we do also many experiment using mutagen, yeah, for rice and for soybean and so on. We can also find some mutated character. Okay? It means that's this is just my opinion that this toothless durian is because of genetic theme from the nature. But do you do something like a artificial mutation? No, so it means that is a natural mutation from my opinion. For the slow growth of durian, genetic also possible because of the something happen in the genome, uh, especially for the growth. It means also inhibited the growth, also possible. But the other one, that's the growth and the development of the plant depend on to beside genetic is the environment. Yeah, if we can maximize the environment, it means we can also accelerate the growth. The environment is the land, the climate, and so on. But if we already treat the plant using optimal environment cannot grow, it's also possible. That is also genetic thing in this character. Thank you, Professor Chan. Maybe next we can share each other for the video side.
Thank you very much, Prof. Vidi, for your answer. Uh, can I add something to that, Prof. Vidi? Because I'm interested when you mentioned mutagens, Prof. Vidi. Have you ever tried to use probably colchicines to duplicate the chromosome number? Have you ever tried? And what is probably want to share some results or experience with that?
Okay, the question is, I also now is my project. I have a project to haploid breeding to increase the chromosome from the wild banana, pisang liar, pisang monyet is cannot be used, cannot be used, can not edible also because a lot of seed in the fruit, so we cannot eat it. Only monkey eat it. Now I conduct experiment to increase the chromosome, and then we got it. We got the doublet chromosome plants. Now it's already growing, and now we are waiting for the reproductive stage, flowering and also fruiting, and we want to know also how about this character of this plant. Afterward, if possible, I will do crossing with diploid plants like uh, Musa balbisiana or Musa acuminata to make a triploid plant. Triploid plant is the modern banana, edible modern banana. Like this is also triploid plants. This is but now is in progress. In my project, we got funding from Kemenistek Dikti.
It seems very interesting. I'm waiting for the results, Prof. So, moving on, is there any more questions from the audience? Probably, okay. Have to see the online. For the online participants, you are welcome to raise your hands if you have any questions.
Hello, I'm Sham from UITM. My question goes to Prof. Asri, right? Regarding seismic, right? If I'm not mistaken. And my question, I noticed that a lot of prediction result from your research, and then I would like to know, and how do you quantify your predicted result? And then how much you can consider it as accurate? This is my question.
Prof. Andri? Oh, I'm sorry. Prof. Andri.
Yeah. So far. Sorry, I didn't hear clearly. Sorry, could you?
No. And I observe from your presentation slide, you research, you have a mix between experimental result and prediction, right?
No prediction. No.
And then I saw a lot of colors, some sort of that.
No, color means the depth of earthquake.
Oh, this is
Yeah.
taken from experimental result. And then,
Just the data from the observation,
oh, okay.
not from prediction, not from experiment. Just we put the seismograph
Yeah.
instrument, and then we record the shaking generated by the event all the time.
Okay, understood.
Yeah.
Observe result. No.
Observe.
Okay,
Yes.
thank you very much.
Thank you.
Thank you very much. Any more questions? We have time until 12 o'clock before we go to lunch break. So probably I would like to comment on some of the keynote speakers. First, I will start from Prof. Andri. First, please correct me if I'm wrong because your presentation is very interesting, but I'm struggling to understand because I am not a geomagnetic scientist. But one of your figures shows, I think, there is a prediction of the declining of the forest oceanic crust. Is that good?
Yeah.
It seems like it's decreasing, and is that a gradual event? Is it happening now? And what is the prediction until years to come? Would it keep? What is the limit? Will it keep going down? And what is the effects towards Lombok Island? Would it still be safe to be living?
Actually, it had been occurring since a long time ago in the geological time scale. So actually, of course, if we have something like plate motion relatively motion each other as consequences, actually we have something like a potency of the seismic occurring in the region. So I mean the mitigation, education, and disaster preparedness among society, government, and all stakeholder. I mean, it must be ready.
Yes.
Yeah, something like that. So that's all. Thank you.
Yeah. So based on my experience when I was in Japan, we have a practice when earthquake occurs, and that we did not have in 2018. So when everything occurs, all the young people went out first. In Japan, that is not allowed. In Japan, we have to prioritize the elders and the mothers and the babies. So that's the SOP, the SOP. And I think that is very important for.
Yeah, actually, no, actually, from BNPB, from BMKG, and also the other institution already published or released about what we should do if the uh, uh, earthquake happen and then the strong motion felt. There is something like, I mean, earthquake early warning, something like that. So it's important to educate from the, I mean, from the family, start from the kindergarten, elementary school, senior school, and also university education about the mitigation, preparedness, and what we should do if the earthquake occur. Thank you.
Thank you very much, Prof. Andy. Okay, I would like to continue my comments next to Prof. Widi. I think I'm interested in the variant Durio Gutagensis. You mentioned in your presentation that it has no alcohol content. Can you explain probably the physiological process? Because in my understanding that for a fruit to get ripened, it should have alcohol content, even it's 0.%.
Thank you very much for the questions. You know that we can see also as a comparison is a montong durian. yeah, montong durian, and also Durio cotejensis, also local Durio jubitunus. The alcohol content is produced from the fermentation process. Fermentation process, especially after if in the climacteric process in the fruit, carbohydrate will change to the sugar, and sugar, if fermented, will be the side. The result is one result is alcohol or something like that. In the local durian, usually because of the insect, yeah, insect go through to the fruits. Sometimes they bring also some not fungi, yeah, fungi or camir, yeah, fungi also bring to it and do some fermentation and produce uh, uh, alcohol and smell very and then ester or something like that the growth process. But for the Duliacuti changes, they have very thick full ya, skin, yeah, very thick skins, and until uh, until rib they don't open. It means that's the possibility to do like a fermentation is very low. It also happen in durian montong. I've also the shelf life also um, length, long, yeah, longer than the other. You can do this. This is in my opinion like this. So if among the durian variety also, we can also find which this one is very strong. Yeah, durian matahari is very strong fragrant because after after rip they directly to break the skin and uh, fermentation directly have local variant, very strong skin and still yeah, it doesn't have alcohol and not fragrant also. This is in my opinion.
Thank you very much for the explanation, Prof. Vidi. And oh, okay, Prof. Saja wants to comment.
Sorry, I forgot to ask one question that very common question about durian, Prof. Widi. Is there any relation between durian and medical? There maybe many people just say uh, the durian has correlation with the high tension blood or cholesterol or so on. According to your experience as an expert of durian, is there any the data that can show us that durian can cause the blood cancer or cholesterol? Thank you.
Langsung ya. Thank you very much. Very interesting question. Many people think that if eat durian will increase the blood tension or something like that. Just simple high carbohydrate high sugar. If there is a fermentation, high also alcohol. It means that a lot. If we eat a lot durian, it means that we also like consume alcohol also. How about the effect of alcohol? Maybe also increase uh, the blood tension. But for the cholesterol, it's different things. Cholesterol that influence the blood tension, especially mostly is from animal cholesterol, pitosterol. This not dangerous for our health. Pitosterol is not dangerous for it. This need is good for our health. But maybe the problem is the alcohol and also the if we eat a lot also if we eat something quite a lot also make a problem with our body also. This is my opinion. It means that's the conclusion is I don't think that durian is bad for our health. I think this is my especially if we make a correlation between durian and cholesterol. It will not relate directly. Thank you very much.
Thank you very much, Prof. Vidi. Very interesting question, Prof. Satya. Yeah. So I would like to add my comments also to Prof. Roleda, who's joining us online. So a long time no see, Prof. Mikey. It's good to see you. You think it seems you're doing very healthy and well.
Yeah, thank you.
I'd just like to ask, you did a lot of kerajinan screenings from your specimens. You focused on only the yield and viscosity, because sometimes here the industries always require us to do tests on the gel strength. So do you think for initial screening, should we consider the gel strength, or we can just focus on the yield and viscosity?
Okay, thank you very much. That is a very good question. Indeed, no, for
I'm sorry.
hello, do you hear me?
Prof. Mikey, can you wait? There's no sound.
Okay.
Hello?
Hello, do you hear me? Hello.
Uh, yes. Wait, the sound is not coming out.
Hello, hello. Hello.
Okay.
Hello.
My question to Prof. Widi, how or is there any method to measure ripeness of durian? For example, we can use natural frequency to measure to measure the ripeness of durian. And then from the frequency, we can determine the level of ripeness of durian. This is my question to you. Is it possible?
Uh, Sorry, I cannot get the point. How to measure
Ripeness.
ripeness?
Yeah,
Okay, ripeness. Thank you very much.
you're welcome.
Usually, if we want to buy durian, we have to look at which one this already ripe or not. usually very simple, very simple way is to hear knife. We can hit maybe sometimes if the hit if the sound is apa ya yang ngebass itu apa yang lebih deep deep sound, it mean that is because there is a space because of if the durian is going ripe, it means there is will be a space in the durian fruit. That's why the sound will be heavy. But if not ripe, it means that the space is very it means that no space because it's full with fruit, with raw fruit, and the sound will be very light or something like that.
So, in terms of natural frequency, are you suggesting that we can use natural frequency to determine the ripeness of durian?
Use what?
Natural frequency. Frekuensi alami. Yeah, the unit in hertz. Natural frequency.
Natural frequency?
Yeah.
Yes, this is not a natural frequency, but this is
No, there is a research. They use natural frequency to measure ripeness of durian. This is my question to you. I'm asking you whether we can use natural frequency to measure ripeness of durian. This is my question. Thank you very much.
okay. It also possible, very possible also if we can use sorry, I didn't get the point. Also, if we can use like equipment or tool to measure the frequency, I think it's also very good, like good finding or good innovations. Yeah, good innovation. We can measure the frequency. If the frequency is very high, it means this raw. At very low, it means rip or something like that. Then we can make like a quantification or something like that.
Yes, Prof. Thank you very much.
Yes, it's good idea.
Yeah,
okay.
yeah. Thank you.
Yeah.
Thank you very much. I think we have to go back to Prof. Michael.
Hello, do you hear me now?
To the previous question.
Do you hear me now? Hello, hi, hello.
We can hear you clearly. Thank you.
Do you hear me? Could you hear me?
Yeah.
Dok Eka?
Yes, we can hear you, Prof.
Okay, thank you.
We can hear you.
Okay, thank you. So that was a very good question, no? Because when we are studying Capophycus alvarezii, or the trade name, what we call as cotonai, this is mostly used for certain application that the gel strength is the more important thing. So Kappa carrageenan is known to really the application is for it to as a stabilizer, no? So yeah. We started looking into the carrageenan yield and only viscosity for now, but definitely it is important to also look at gel strength. And indirectly, we could say that the more viscous the solution is, it will be has a higher gel strength. So there is a direct relationship. So indirectly now we could say that our sample with higher viscosity would also exhibit higher gel strength. But definitely, we need absolute data looking at gel strength.
Thank you, Prof. So, in terms of when we want try to explore source of carrageenan, should we focus only on spark carrageenan, or does IMSA carrageenan also have to get some attention?
Yeah,
Because as you
yeah. also a good question, because this will also depend on the application. Because, for example, some of the application, they would require iota carrageenan, then definitely the production of spinosum or Eucuma denticulatum should also continue. And I have discussed with different processors here in the Philippines. For example, with Schemberg, I have a good cooperation with this processor, and they actually do some mixing. So depending on their client, they want only a very low gel strength, low viscosity. So they would mix different types of carrageenan. So mixing kappa with iota, and then they will get a certain consistency, a certain quality that is very specific to a certain application. So at the moment, I am also actually doing some kind of a collaboration to look into the industrial application of beta ficus, because as we know that beta ficus produces a mixture of beta carrageenan and kappa carrageenan. So and there are enough biomass in the wild, and we are also examining the cultivation technology for beta ficus. And then hopefully, if there is some application, then we have more species for cultivation. No? So not only now the kappa ficus and the Eucuma, maybe eventually with beta ficus. So it's more of addressing the what the commercial need are. So it's more of customer driven, the application.
Oh, okay. Thank you. Seems nobody's asking, so I would like to continue. So based on the all the methods, it seems like the alkali potassium hydroxide method seems to be producing the highest yield, if I'm not mistaken.
Mhm.
And what is your opinion if this method is used in the industrial scale in terms of green industries practice? Because this is I am actually using this as an example because there is an industry in Indonesia. I don't want to label the region to cause any conflict of interest. So there's an industry in Indonesia, carrageenan. They don't produce carrageenan, but ATC, so alkali treated carrageenan.
Yeah.
And then use potassium hydroxide, absolute potassium hydroxide, and they decrease it to 60%. In my opinion, it still has effects on the environment, Prof.
Yes.
Mikey. So
Yeah.
what is your opinion on that?
That is very correct, and I completely agree with you. No, for in terms of producing sustainably and the green label. No, because definitely the carrageenan yield and also the quality improve with alkali treatment, but using different kind of alkali at an industrial scale, this has environmental concern in terms of the disposal of the chemicals. So I mean, it's more of like now putting my hat as an environmentalist or putting my hat as a processor. So I think we need to have a balancing act here, and I think the best way to go is to really find some kind of a disposal system or cleaning up system that the wastewater or the rest alkali solution will not be indiscriminately thrown away in our sewer or in our disposal system. So in the end, it will be like a cost benefit analysis that we will do, because without alkali treatment, what we call the native extraction, it still give you some kind of a lower quality or lower yield, and then if you put some alkali, it improves a lot. No, so in the end, it's more of assessing or having a technology wherein we could minimize the negative effect or completely, I mean, eradicate the negative effect of the alkali wastewater.
Yeah, thank you. I agree with your comments, but partly probably from my because yeah, I think in this region they have this industry going on, and they are struggling to grow again good seaweeds, and yeah, they are looking for new seedlings as an opportunity, but it seems it's not the seedlings problem. There's something wrong with the environment, in my
Yeah,
opinion.
yeah, yeah. Yeah, I also agree with that observation, no, that uh, we always thought that it might be the quality of the seedling is the problem, but unfortunately, due to some of the contaminants in the water, that is basically affecting the growth of the seed stock or the plants that we are planting. And we also notice this, or we get some reports from our seaweed farmers wherein there are some fishermen engaged in some illegal activities using chemical to stun fish or using chemicals to clean up their fish ponds. And then when the wastewater goes out, this goes to the seaweed farms and all the seaweed die. So there's a conflict of use of the coastal environment in terms of fish pen, fish pond, or using some chemicals to stun fish or in the intertidal flood. No, so yeah, this should be really looked into.
Thank you very much, Prof. Florida. So, lastly, I would like to welcome again from the floor. Is there any more uh, questions? I'm hoping there are some questions also from online participants, but it seems also there's no questions from our online participants. Okay. So because we are up, we have our time is up now for the uh, first plenary session. I want to invite everyone to give a big round of applause to all our keynote speakers. Yes, and hopefully, if everyone interested to join collaboration, you are welcome to contact the keynote speakers privately via Zoom chat or probably give an email in their presentation. Thank you very much for joining our morning session today. So probably the lunch will be okay. The location of the lunch will be explained by our master of ceremony. And once again, I would like to thank the keynote speakers for the attending
Thank you very much.
our conference today. Wassalamualaikum warahmatullahi wabarakatuh. Thank you, Mikey.
Thank
All right,
you.
thank
Thank
you
you,
so
Dr.
much,
Bye.
Dr. All right, thank you so much, Dr. Eka Sunarwidi Prasetya, for being the moderator of this discussion, and also our amazing keynote speakers. Thank you so much.
All right, ladies and gentlemen, we would like to hand over souvenirs first to both of our keynote speakers. The first keynote speaker, Professor Dr. S.J. Insinyur Andri Dianugraha SSE MSE, and also to the keynote speakers to Professor Widi Sunario SPMSE PhD. And in this occasion will be given by Professor Sukarto MAGR as the head of LPPM, and also Professor Insinyur Puan 2023. So please come forward to the stage to give the souvenirs. All right. Maybe Professor Sukartono would like to hand over the souvenir to Professor Dr. Insinyur Andri Dianugraha first. All right, ladies and gentlemen, let's give a round of applause. Thank you so much, Prof. Yeah, and we will have photo session, of course. Thank you, Prof. And the second keynote speaker to Professor Widi. All right. Okay. Souvenir first. I'm so sorry. Thank you. And the second souvenir will be given to our keynote speaker, Professor Widi Sunario, SPMSD PhD, and it will be given by Professor Insinyur Shari. Yeah, we will take photo. And also we will hand over. Ladies and gentlemen, let's give a round of applause, please. And of course, the souvenir. Thank you so much, Prof. All right, ladies and gentlemen, we will immediately have our lunch break. Yeah, but before that, I would like to inform that for the participants for on-site parallel session will be conducted at 3 rooms. The first room is Selaparang room. The second room will be in Mayura 1 room. The third room will be in Mayura 2 room, and these rooms are located on the ground floor, 1 floor below the ballroom. So the parallel session will be conducted below this ballroom, right? And of course, we will have our lunch break session. For the lunch break, the keynote speakers and also the invited speakers will go to the upstairs, the restaurant upstairs for the lunch break. And also for participants, you can go straight to restaurant on the same floor. And we will be back again at 1 p.m. sharp for the parallel session. Please go straight to your room, and thank you so much and enjoy your lunch. One more time, I would like to inform that for participants, you can go straight to the restaurant on the same floor to have your lunch, and also for the keynote speakers and also the invited speakers to please go to the upstairs restaurants. And we will have the parallel session on the first floor below this ballroom to the Slaparang 1, Mayura 1, and also Mayura 2 rooms. Thank you.